Hello again.
We have a few people in the waiting room already. Are those the presenters? I think I saw Alex Smith.
Yes, all of them are presenters. Hello, everyone.
My thesis project is on least Tern disturbance responses to human activity on Hatteras Island in North Carolina. Least Tern.
I'm going to try sharing my screen. There's one presentation that I have to share. So I'm just going to make sure that that works. Can you see it? I can't see you, so I need someone to tell me.
Yeah.
Okay.
My thesis project is on least Tern disturbance
Can you hear it?
responses to human
Yep.
activity on
Okay.
Hatteras Island.
Was good.
Great. If anyone wants to check their audio or anything, we have like 15 minutes still, so.
I'm actually going to switch computers real quick. So I'm going to log off and be right back.
Sounds good.
I don't have any audio in my presentation, so I'm not too concerned about that, but.
Oh, I mostly just meant your mic.
Okay, cool. Can you hear me okay?
Yeah.
Yeah.
Okay, cool.
And I just want to check. How do you pronounce your name? Islet?
It's Aylet.
Aylet. Okay.
Yeah.
Glad I asked.
Thanks. Can you guys hear me okay?
Mhm.
Yes. Mhm.
Okay, cool.
Does my audio sound okay?
Yes.
Yep.
Then I'm good.
And are you still there? Never mind.
I am. I just
Okay. No worries. In the last session, I think my internet connection became unstable, and so the audio cut out for a moment. Riley and I are both calling in from the same place. So I'm just going to assume if nobody puts anything in the chat that the audio is still working.
I am happy to let you know if anything happens.
Okay, cool.
I'll be right back.
Is everyone able to hear me?
Yes.
Awesome, thank you. A quick question, if I may. When is the first question session for the first piece of lightning talks?
Yeah, I just emailed you back. It
Oh,
does
okay,
start at 3:55 East Coast.
Okay, perfect. And that's for, you said 10 minutes?
The next talk actually doesn't start until 4:20, so
Oh, okay,
we have like 30 minutes
okay,
of time to fill.
okay, great, thank you.
Yeah.
And Jenna, sorry, you might have mentioned this already. I was a little bit late. Sorry about that. But do we just hit the share screen button when it's our turn to speak?
Yeah. If you want to practice and make sure that it works okay, you can go ahead and do that.
Okay, I will give it a go. It's still disabled at my end.
Okay, try now.
Good. There we go. You see that okay?
Yep.
Beautiful.
All right.
Actually, Jenna, can I go ahead and try mine just in case?
Yeah, absolutely. That's what this time is for. Yep, looks good.
Cool. Awesome. Okay.
I'll have a go too.
That looks good. It's not in full screen. I'm not
No,
sure if you
yeah.
wanted that.
Yeah, I can have a go, but yeah, I think that's okay.
Yeah.
Can I also try?
Yeah, absolutely. Looks good. Oh yeah, you can hear it too. Oh, that's so fun.
Cool. Thank you.
Yeah. And I'm just gonna make you a co-host just in case.
Yeah.
Or maybe, actually, Riley, you might have to do that. I think you came in first, so.
Okay.
So if you go to participants and go to Anna Valerie Moore, you should be able to make co-host.
Okay.
There's a snowstorm coming through here, so I'm nervous.
No worries. I'm happy to step in if you guys
Disappear.
here to the ether.
I believe I just made you a co-host, Anna.
Yeah, I can see it.
Okay, cool.
Where are you? There's a snowstorm?
Seward, Alaska.
Oh,
Yeah, we're gonna get maybe 13 inches of snow tonight.
wow.
Yeah.
Oh, Jenna, I think I'll just make you host.
Oh, great.
There.
And I will remake you co-host. Hello.
Hi.
Welcome, everyone. People are still coming in, and we're just going to get started in another minute.
Jenna.
Did someone say my name?
Yeah.
Oh, hi.
If there's any questions in the chat, should I wait for the question bit, or should I just answer them when
Yeah.
they come through?
Yeah, I think because we have 30 minutes between the first and second part, that we might as well save them.
Okay. Yeah.
Yeah. And then if there's any that we don't get to, then yeah, just answer them in the chat. All right, so I think for the sake of time, we'll get started. So welcome everyone to the Waterbird Society virtual conference. This is the lightning talk. So the way that this works is it's 5-minute back-to-back presentations. And then there are 2 sessions. The first session is the student work in progress session. And so that ends at 3:55, and after that we'll have about a half an hour for questions for the first group of speakers, and then we start again with general lightning talks starting at 4:20. If anybody has any questions, you can go ahead and put those in the chat, and then when we get to the question session, then we'll go through those questions. All right, so I think we can just get started. Up first we have Matthew.
Okay. Thanks, Jenna. There we go. Hi, everybody. Thanks for coming today. I'm Matt Leggett. I'm a first-year PhD student in the Davron Lab at the University of Manitoba. And today I'm going to be talking about my early findings as I investigate personality in razorbills and the effects that this may have on their fitness. All of this work was carried out this summer on the northeast coast of Newfoundland on James Island, and you can see that shown by the red dot on this map. Animal personality is where animals show consistent between individual differences that persist through time and across contexts. And you can see that sort of shown by this example data on the right, where each line, each color represents a different individual. And you can see how these differences continue through the time period. There are 5 main studied personality traits: boldness, aggression, sociability, exploration and activity, and boldness is the most commonly studied. These differences are common throughout the animal kingdom and have begun to be identified in seabirds. And boldness has been the sort of primary trait that's been studied in these in seabirds. So look at this in razorbills. We carried out 3 tests of the boldness trait on 30 individuals this summer. And these 3 pictures across the top illustrate these 3 tests. The first test was we measured the response to extraction from the nest crevice. So this picture shows the typical razorbill nesting habitat that we have. And we measured the response to withdrawing the animal from these crevices between the boulders. This middle picture shows a razorbill in a pillowcase, and we measured response to 5 minutes of restraint in this pillowcase. And finally, in between these 2 tests, we were handling the birds for routine procedures such as taking feather samples and deploying GLS tags. And the 2 or 3 researchers that were present during this stage rated the birds on their boldness. These 3 plots show the results of these 3 tests, and they illustrate both the wide range of responses within each of the 3 tests, and also that these 3 tests significantly correlated with each other. So the boldest bird in one test was also the boldest bird in the other 2 tests. And in addition to this, we looked at the consistency between the raters, consistency between the ratings by the researchers, and found that these were also highly consistent. So we got Kendall's concordance values of greater than 0.8. And a value of 0.6 is typically considered strong agreement. So taking all of this into account, we carried out a principal component analysis, and this was used to combine the scores from all of the 3 previously mentioned tests into a single boldness value for each of our 30 birds. Principal component 1 explained a large amount of the variation and was highly correlated with all original variables. And you can see these boldness values over here on the right for our 30 birds this summer. And this again illustrates the wide ranges of response that we found in our 30 birds. So moving on into next summer and beyond, we will be carrying out the same tests on as many of these 30 individuals as we can, so that we can test consistency over time by using repeatability analysis. And that will hopefully allow us to take this early evidence of personality in this species and turn it into proof of personality in this species. Finally, we looked at the breeding success of these individuals by monitoring nests every 2 to 4 days throughout the breeding period and combining this with data such as feather development of the chicks, chick growth rates, and things like that to attain a best estimate of each individual's fledging success for this year. And we found that successful breeders were more bold than failed breeders in this summer, but it's important to note that the sample size for this is very small. So this is something that I'm hoping to improve on in future years, and also to see whether by carrying out this on the same individuals in multiple years, to see whether this relationship changes depending on prey availability in the study area. I'd like to finish by saying a quick thank you to everybody that helped this summer, so the members of the Davron Lab and the crew of the Lady Eastern, and thank you for listening, and I look forward to any questions.
Thank you, Matt. And as I mentioned in the chat, these are 5-minute back-to-back lightning talks, so we'll get to questions at the question session. Excellent presentation, and we're going to be moving right along to Erin.
I'm sorry, can you hear me and see my screen okay? Hello?
I think so. The audio is a little funny,
Okay.
but we can hear you.
Okay, you can hear me?
Yeah.
Okay, great. First of all, good afternoon. I'm really happy to be here to share my research with you all and to give feedback on my project. My name is Erin Gallagher, and I am a graduate student at the University of North Carolina, Wilmington. My advisor and co-author is Dr. Raymond Danner. And today I'm going to be talking about estimating nest productivity and identifying sources of nest failure in Least Tern colonies in the Outer Banks of North Carolina. Okay, so my study species is the Least Tern, or Sternula antillarum. Least terns are colonial water birds that nest on the beach. They're ground nesting birds. Least Tern colonies can vary greatly in size, shape, density, habitat type, and species composition. Least terns are a species of special concern in North Carolina, mainly due to the loss of their nesting habitat. As such, the protection of these nesting sites is of great interest to both federal and state agencies throughout North Carolina. My work is part of a cooperative agreement with National Park Service at Cape Hatteras National Seashore. The overall project objective is to assist the National Park Service with evaluating the efficacy of their existing protection protocols for nesting least terns, and to eventually recommend any changes to the current protocol if necessary. We are going into the 3rd year of this long-term project, and it has many different components to it. However, today I would like to take this opportunity to get some feedback on the design of my data analysis, particularly in regard to estimating the survival rates for least tern nests. So we monitor nests in Cape Hatteras National Seashore and Island National Wildlife Refuge on Hatteras Island, North Carolina. Here is a quick summary of the work we've done thus far. 2019 was a preliminary study year, and then in 2020 and 2021, we were able to intensify our monitoring efforts to include more colonies and more nests. Data collected include nest fates for each of the nests monitored. I will be taking these nest fates and using them to estimate daily survival rates for the least tern nests using survival analyses included in the RPAGRISH RMARC. Daily survival rate is considered the probability that a nest survives from 1 day to the next and so on. And I plan to use Akake's information criterion to select the best models. And then to identify biologically important covariates, I will be using beta coefficients to estimate the covariate's effect size. Covariates of interest include nest age. So least terns typically incubate for 21 days or about 21 days. And we try to identify nests at or near initiation so that we can follow nest fate throughout the entire incubation period. I'd also like to identify any temporal variations both within the nesting season and between different nesting years. And I also plan to consider management practices since the National Park Service at Cape Hatteras and the U.S. Fish and Wildlife Service at Pea Island have different protection protocols for least terns. I like to look at different colony characteristics like size and density, as well as multiple biotic and abiotic factors. Biotic factors include predators like mammalian predators, ghost crabs, and avian predators. We look at direct depredation events, as well as any predator presence near or within the colonies. Least terns will also interact with each other and other nesting species that are nesting within or near their colonies, so the presence of other species and their interactions are recorded. There are also human impacts that we record, such as direct interactions like flushing due to people getting too close, and this is a topic that my project partner Alex will be presenting on right after me. And then indirect impacts like proximity to roadways and villages, which can influence other factors like predator presence. In this picture here, you can see Least Tern incubating a nest right next to NC12 with a truck right behind it. Abiotic factors include weather events such as tropical storms, tidal events, and any abnormal weather that leads to nest loss. I also record weather data over the course of each nest incubation period, such as average daily temperature, wind speed, rainfall, and solar power. I am also considering the use of Bayesian hierarchical models. This is an approach that I haven't really explored fully yet, but I'd love to hear if anyone is familiar with this sort of analysis. I envision using it for age-specific nest survival rates. And then I am also in the process of developing my methods to determine chick survival. So we were able to ban Least Tern chicks this past summer with field-readable identification bands as well as site indicator bands. And then we also were able to use some radiotelemetry tracking at one of our colony sites. So if you have any advice regarding mercury site survival modeling, please let me know in the Q&A. And just a second to thank everyone who has helped me in this project over the last 2 years and made this project possible.
Thank you so much. Again, we'll save the questions for the question section. So if you have any questions, you can go ahead and put those in the chat. I am going to be sharing my screen for this next presentation.
My thesis project is on least tern disturbance responses to human activity on Hatteras Island in North Carolina. Least terns, the study species, are a colonial ground nesting water bird, which in this case are being studied in both Cape Hatteras National Seashore as well as Pea Island National Wildlife Refuge. These birds are a species of concern in North Carolina and are viewed as somewhat vulnerable. And due to this, there are protections put in place by both Cape Hatteras and Pea Island to limit adverse interactions of these birds, specifically while breeding with humans. Typically, they take the form of a line of signs around these protected nesting areas with string and flag between. And in Cape Hatteras, they specifically establish a 100-meter buffer between this line of signs and the nearest least tern nest or chick. These buffers are effectively being tested throughout this project to determine if they should stay the same or be minimized or perhaps should be bumped out further. In order to test to figure out the best way to create these buffers and maintain them, I have 3 null hypotheses looking at human disturbance. The first is current buffer designs protect least terns from behavioral disturbance. The second is the type of disturbance will not differentially affect least tern behavioral response. And the third is that least terns do not habituate to disturbance factors throughout a season. Last summer, I completed opportunistic protocols to establish a baseline for experimental data collection this summer. And so this summer, I plan to use more manipulated protocols, looking at least tern disturbance response to specifically pedestrians and off-road vehicles, which are allowed on the beaches of Cape Hatteras National Seashore. I plan to look at differences between direct and tangential approaches or passes, and wish to quantify frequency and intensity of these disturbance responses. I hope to do 3 separate treatments, or treatment times, I should say, morning, afternoon, and evening, to limit any potential bias there. And I plan on using 10 approaches or passes with a pedestrian or truck for each of these treatments. I hope to use at least 4 least tern colonies of varying sizes. And in this case, within a week, 2 of those will be exposed to treatment for 3 days, rotating their treatment time period each day. And then the other 2 would be exposed to treatment for the next 3 days, and we would switch off like that. In order to gather this data, a spotting scope would be trained on a single target nest, and 1 or 2 cameras would be trained on the group of nests around this target nest to try to capture any ancillary disturbance responses within these colonial birds. The response variables I hope to gather are flight initiation distance, which is defined as the distance between the bird when it took flight and the source of the disturbance. Agitation initiation distance, which is similar, except it's the distance between the source of the disturbance and the bird at the time that it became agitated or showed some sort of agitation behavior, as well as flush duration and percentage of the reacting terns within that target area of the cameras. So a bird could show agitation, and then flight as the truck or pedestrian moves across, or perhaps just no response. In order to control for possible biases within this data, I hope to complete control observations for 10 minutes before each treatment and allow birds to settle between each pass or approach. These treatments will occur during both weekdays and weekends, switching between colonies to make sure that all colonies are measured in case there are differential weekend and weekday disturbance responses due to the general fluctuations in human activity from visitors on the seashore. And I hope to use variable clothing colors and possibly even masks for each treatment period with pedestrians, as well as variable coloration on our vehicle as needed so the birds can't learn the faces or vehicle itself used for these treatments and react any differently than they might a random visitor's vehicle or pedestrian. I would like to thank National Park Service and specifically Cape Hatteras National Seashore for their help and funding for this project. U.S. Fish and Wildlife Service, specifically Pea Island, for their help in logistics throughout our field season, as well as the UNCW Danner Lab and my graduate committee.
All right, thank you, Alex. And we are going to move right along to Islet.
Okay. Hi, everyone. My name is Ayla Litford, and today I'm going to be talking about my master's work at Louisiana State University, which is assessing the value of created marshes for wetland birds in southeastern Louisiana. Sorry about that. As you all know, wetland loss is threatening waterbird populations around the world. Here in Louisiana, we lose an average of 100 square kilometers per year, which is about a football field every single day. And in order to combat this extreme land loss, Louisiana has initiated a coastal master plan, which designates funds for marsh creation and restoration. But while many of these projects have been successful at just putting land back on the landscape, little is known about how wetland dependent wildlife respond or if created marshes are functioning similarly to natural marshes. So therefore, our study is looking at these created sites and seeing how they are good for wetland birds. So our objectives are to compare abiotic characteristics such as hydrology and elevation, and biotic characteristics such as vegetation and bird presence between created and natural sites in the hopes of informing restoration strategies. Our study sites are located all across southeastern Louisiana. This map shows 6 created marshes that were surveyed in 2021. So we just finished the first of 3 field seasons, and we will be adding 5 more created sites for 2022 and 23. And for each created marsh, we also surveyed the nearest natural marsh to use as a reference site. For our methods to compare water levels, flooding frequency, and inundation time between sites, we installed water level recording devices shown on the left. And we installed those at all of our created sites. And we also conducted vegetation surveys and point count surveys for all birds, including a callback survey for secretive marsh birds. So our preliminary results showed that created marshes varied widely in hydrologic regimes and vegetation communities, but many of the created marshes had overall lower water levels than the natural marshes, and therefore they had a more upland and shrubby vegetation community. These graphs show water levels throughout our field season, with the red line being ground level and the blue lines being water levels. So the site on the left had extremely low water levels, except for short periods of higher water, most of which were precipitation driven. And this site therefore was extremely shrubby. But the graph on the right is another created site that had very high water levels, and they were a lot more variable. And this more closely resembled a natural marsh, and it was dominated by three-corner grass. So what does this all mean for the birds? Our results show some trends that certain species may be selecting for or against created sites. Common gallinules were our species that were most commonly detected overall, and they, as well as purple gallinules, were detected much more at natural sites than created. And we believe this is due to more water availability at the natural sites, since gallinules need shallow water and ponds for feeding. Seaside sparrows were also detected much higher at natural sites, and this makes sense since sparrows are grassland specialists, that they preferred the natural sites over the shrubby created ones. But on the other hand, common yellowthroats were detected much more at the created sites, most likely due to more shrubs for nesting. And most surprisingly to me, king and clapper rails, which we recorded as cling rails, were found more at created sites as well. So we're really excited for what the next 2 years will reveal, but we believe that current trends and further investigation will show that site-specific hydrologic processes are major drivers of waterbird communities. Both created and natural sites vary greatly, and creating marshes at the right elevation that allows for flooding and establishment of emergent vegetation is the key to creating suitable habitat for waterbirds. So just a quick thank you to all of our collaborators, especially our funding agency, Louisiana Department of Wildlife and Fisheries. And thank you all for listening. I look forward to your questions at the end, and also feel free to email me with any questions or feedback.
Thank you, Ayelet. And we're gonna move right along to our last presenter, Kay.
All right. Right. Hope this works now. All right, I'm gonna start. Hello, everyone. My name is Ke Zhang. I am a PhD student at the University of Florida. Today, I will present you part of my PhD study, habitat use of white ibis and tricolor heron in the northern Gulf of Mexico. Some background here. Northern Gulf of Mexico, it's an important region for wading birds. Many species migrate here to spend their winter, and several islands here are used by wading birds as their breeding sites. And as most of you may know, in 2010, an infamous oil spill incident occurred in the northern Gulf of Mexico. This incident seriously damaged local wildlife population and the ecosystem. So wading birds are among those animals which suffer a lot from this incident. However, when estimating the damage caused by this incident, people realized that there was not enough baseline data on wading birds in this region. Very limited information is known about these birds' movement ecology, especially not much known about their habitat use. And this information about habitat use can help identify critical habitat that need to be preserved to serve the purpose of population restoration. And for wading birds, most of the information we know is obtained from their breeding season. Therefore, there is the need to understand their habitat use in non-breeding season. So why we should study habitat use in non-breeding season? There are at least 3 reasons. First, large proportion of for birds, a large proportion of annual cycle is in.
Sorry to interrupt. Can you make it full screen?
Oh, all right. So how about now? Okay.
Yeah, you're good. Thank you.
All right. Sorry about that. For first reason is for birds, largest proportion of annual cycle is in non-breeding season. And second, the condition of bird before breeding season affect their reproduction success in the breeding season. So understanding the habitat use before breeding season helps explain the condition of bird. And third, because before wading birds reach their sexual maturity, they are either yearling or can be called subadult, and they may not join the breeding colony in the breeding season. Therefore, for a long time, this group of birds have been neglected in the habitat study. To build up knowledge about wading bird movement ecology in this region, so we choose tricolor heron and white ibis as our study species to investigate their habitat use in non-breeding season. So these 2 species represent 2 typical wading bird groups. Tricolor heron mostly use fish as their food and are more solitary when foraging and have typical migration behavior. White ibis mostly use crustaceans foraging in group and have nomadic behavior, which means they often shift distribution based on resource availability. I am especially interested in 2 topics. First, because yearling and subadults are still under development and their nutrition demands can be very different from adults. I am curious if yearling, subadult, and adult birds use habitat differently. And second, I am curious that compared to nature habitat, if agriculture and aquaculture land, which provide additional food supplies to birds, attract these 2 species in non-breeding seasons. Our study sites are near shore islands in coastal Alabama. In the last 2 breeding seasons, we captured birds and deployed 80 transmitters on them to obtain location data. And for the next step, I will obtain land cover information from National Land Cover Database for habitat use analysis. And all birds are captured and handled under the University of Florida IACUC. So my next step work is to analyze data, but there are at least 2 major challenges now. First, because our study crossed multiple stages in bird life history, it is hard to define resource availability when there are multiple separate non-breeding home ranges for many individuals. And second, our birds cover wide spatial range, and some birds already flew across the Gulf of Mexico. It can be hard to obtain land cover data for the area in the southern Gulf of Mexico. Finally, this project is funded through the Deepwater Horizon Oil Spill Assessment and the Restoration Plan. As I am revising my proposal, I look forward to hearing feedback or suggestions, especially on my method. Here I have my email attached, and as I mentioned at the beginning, it is an ongoing research. I hope we'll be able to share some exciting results in the near future. We have a Facebook page for this project, so if you are interested in hearing more, please follow us on Facebook. And thank you for listening.
Thank you so much. All right, everyone. Now we have the next 20 minutes open for question and answer for all of the speakers that were in the first section of the session. And again, these are students who are all working on their projects. And so feedback and advice is super helpful. I'm going to go through some of the questions that we already had in the chat. Matt, this first one is for you from Kate. How did the 2021 rate in terms of environmental conditions and how did 2021 rate in terms of environmental conditions and nest success? And was there food stress?
Okay.
Sorry, I'm trying to focus.
Thanks,
Oh,
Kate.
I'm sorry. Not for me. Okay, sorry. It's not for me. All right, let me mute myself.
I'll get it out early that this is my first summer on a seabed colony, so there's a little bit of bias here. a little bit of lack of experience on my part, but we did feel that summer 2021 was pretty good in terms of food availability and their success. We had like razorbills sort of fledging at really good weights quite early, and sort of really high provisioning rates amongst the adults, so good size prey for our study area. So we felt it was pretty good.
Great, that's all I was asking. Thanks. It was a really cool project. So good luck finishing up.
Thank you.
And the next question that I see in the chat, and again, if anybody has any questions or comments or advice, you can throw it in the chat or raise your hand, and then I can have you unmute yourself. But the next one that I see is for Aylet. Helen said, really interesting project. Why were the water levels lower in the created site? And is that something that can be fixed easily, or were they designed that way?
It cannot be fixed easily, unfortunately. It's like I said, it's really variable by every site, but the ones that have really low water levels were built at really high elevations, so the water can't get up on the landscape. And unfortunately, a lot of these marshes aren't built with wildlife in mind. They're just trying to get land back on the landscape, and they also have to account for subsidence and rising sea levels. So they tend to build them really high in the hopes that, you know, in 10 or 20 years, they'll sink and kind of be more like a natural marsh. But I'm looking at sites that are already 20 years old and they're still really high and dry. So it is a design issue. And my advisors are actually in contact with engineers and trying to get them to, you know, consider these things when they're designing it. But it's really hard to design a creative marsh. So, but we're trying to get them to kind of think about that more.
Thank you. And while we have you talking, Ariel asked, she said, hi, thanks so much for your presentation. On Long Island, we have a lot of man-made dredge spoil islands where various waterbirds have begun nesting over the years as a result of human disturbance and habitat loss of our barrier beaches. I am currently looking at tern species with a focus on the roseate terns, which historically nested on the south shore of Long Island, but don't anymore. Would you have any insight into choosing best created islands for habitat restoration projects?
I'm not sure if I do. The species that we're focusing on are secretive marsh birds and kind of wading birds, because we don't really have terns or colonial water birds in the marshes where I'm working. So I'm not sure what would be best for them, but I'm assuming it would be different from secretive marsh birds because they need some water, some standing water in the marsh at all times, which is what we're really trying to get the engineers to think about. But it seems like for terns, obviously you don't want flooding for nesting terns. So I can send you some contact information and maybe we can talk about this more some other time. But it sounds like a cool project.
Great, thank you so much. And for our last presenter, Dave Moore asked, did you see any age effects regarding the local and longer distance migrants?
Yes, actually, that's one of the topic we're interested in. Although we haven't done the very formal analysis yet, but we do see like some other people research mentioned the young birds usually don't directly conduct a very long distance dispersal. They will instead they will do some short distance dispersal and stay relatively close to where the breeding site was. But we also see some young birds fly just across the Gulf of Mexico, and so we think age should be playing some role here, but there's no very formal analysis yet.
Great, thank you so much. Next question is for Aaron. Can you tell us what transmitter technology you used on Least Terns?
Oh, sure. Yeah, we use cellular tracking technologies, radio telemetry system, and I actually saw your question on it in my lab, so I just grabbed a little tag so you could see it. They're very tiny. They go right on the back of like little terns, and they have a very small antenna. So we put them on the Least Tern chicks, and this particular way of tracking birds uses a node system, so a receiving node system where you put multiple nodes out. You can use grids for certain setups, but we, for our purposes, put the nodes in like around the perimeter of a colony, and so the nodes pick up the location of these tags, and then it sends it to a sensor station, which then compiles that data and sends it to an online database, which is updated every hour. And we can actually take that data and create like these daily reports that allow us to like look every morning before we go into the field to see where the birds have been hanging out on the beach, so that we can have a better chance of actually spotting them again. But yeah, it's just called cellular tracking technologies, life tech. So it records data night and day. We also have solar tags available, but they can only work at night, or sorry, during the day.
Great, thank you so much. And for our last speaker again, how will you tackle the differing land cover resources available across the study area?
Yeah, that's one of the challenge I just mentioned. I think, yeah, it will be very hard. What I'm thinking now is probably have to check each individual bird and see if there's any like significant pattern. I have to divide it, the whole data set to several periods, and each period there will be like relatively stable resource availability during that time in that area. But I hope I can like hear more suggestions on this part too. If anyone have some better idea, please let me know. Thank you.
I don't see any more questions immediately. Again, if you have any questions or comments or pieces of advice to give any of the students that spoke, you can put them in the chat or raise your hand. Oh, one just came in for Alex. Dave said, I may have missed it, but do you have a control area or low disturbance area in your study? And do you think that is necessary?
Yeah, so that's a great question, Dave. Unfortunately, due to the fact that it's within Park Service and also National Wildlife Refuge grounds, we don't have a control area that will have no disturbance, mostly because beyond our own research project, the National Park Service and the Pea Island National Wildlife Refuge goes through and does their own surveys for these birds, for the least terns. So they are in there part of the time as well. And along with that, while the Park Service and Wildlife Refuge does its best to limit adverse interactions with wildlife, violations of these closed areas, it's kind of impossible to completely keep that from happening. So yeah, unfortunately, we don't have a control area, but there is kind of a spectrum of amounts of disturbance based on where these different colonies exist within these resource protection areas. So some areas are lower than others, which we hope to kind of use to look at some differences between how birds respond in these lower disturbance areas versus higher ones. But unfortunately, there's no area that we can maintain as a complete control without human disturbance.
Thanks. I have a question for you actually. When you're doing these disturbance trials, what happens if there's something else that happens that disturbs the birds, like a predator flies through or the colony or something?
Yeah, so that's a great idea. That'll definitely be something I think I'll have to tease out a little bit more in the field as we're going through it. But I would say at that point, so this past summer, I was looking at opportunistic disturbances. And so we have some data already on how the birds respond to natural sources of disturbance. But I also plan to gather as much data as I can this next summer, even if it's opportunistically. So if it's quite clear that the birds are responding to this natural disturbance over a human-related disturbance, we would still write down that data. But obviously, we would probably have to redo that treatment to make sure that we were getting data only from that single source. So yeah, definitely something we'll have to work on, but we want to make sure that data is clearly coming from that anthropogenic source.
Yeah, thank you. Anna has a really great point she put in the chat that the Waterbird Society Slack workspace is also a great place to touch base with presenters to continue conversation, and she put the link in there. So if anyone's interested in that, definitely head over there. Matt, I had a question for you. What behaviors were you looking at with each of your trials? I know it was a short space to fit in everything.
Yeah. So the extraction from the nest was biting, vocalizing, and struggling. The restraint test was struggling and rate of breathing, and the behavior ratings was a whole suite of things. So biting, vocalizing, struggling, difficulty to take measurements, all sorts of things like that.
So when did you give them like a biting score, or was it kind of like a presence absence kind of thing?
For the first two tests, it was sort of much more sort of quantifiable presence absence, but counts as well. And then the ratings was just like, because we were sort of like busy working on the birds, and it was like between 1 and 4 in the morning and at night. So we were, it was very much just sort of like a best impression based on everything that happened in sort of the 5 or 10 minutes that we were handling the bird.
Oh, sorry. Thank you. I think Riley might have a question. Riley, is that you?
Yep.
Okay.
Let me start my video. Hey, Matt, I was curious if you had any plans to investigate any sort of like physiological or genetic associations with those behaviors?
Not yet. No, not really. Not at the moment.
Thanks.
Thanks for the question, Riley. And I also just want to give a shout out to Riley for being an excellent technical assistant. Thank you so much for your help with this. We have another question for you, Matt. Are you planning on looking at any other of the personality traits, or are you focusing mostly on boldness?
Mostly on boldness, yeah, at the moment. We'll probably touch a little bit on exploration because it can be so come so hand in hand with boldness. And a lot of the GLS work and GPS work that we'll do will be like looking at migration distances and things like that. So we'll probably get into exploration a little bit, but that'll probably be it.
So there's boldness and aggression, and they're kind of related, but also different. So how do you plan on like teasing that apart? You know what I mean?
That's a good question.
I had a similar problem, which is why I'm asking.
Yeah. I'm not sure. I'm gonna have to think on that one a little bit. I guess the main thing we were sort of going with was sort of these sort of forced scenarios for the bird, because it sort of tends to get towards boldness. So the need to escape and things like that. So I guess there is an element of aggression there too. And we may never be able to get rid of that entirely, would be my thought. But we're also looking at the sort of struggles and vocalizing as well. So I hope it's sort of leaning a bit more towards boldness.
Yeah, it's super tricky. Does anybody else have any questions or comments or pieces of advice? Is there anyone with any Bayesian experience that could help Aaron? Or experience with mercury capture? I'm sure that there's someone in here. Or do any of the speakers have any questions or comments for each other? We have 7 minutes until the next presentation. Dave, go ahead. Thank you.
I just wanted to say, like, again, like questions like that, like, does anyone have any Bayesian experience? Or I know I was talking to him yesterday, and he's like trying to figure out how to quantify home range size. There's like multiple methods for doing that. So I think like using Slack, that's like, it's one of its main like good things about it. Like you can just post these general questions, and I'm sure like people that have experience or so forth that'll like respond. So like I just encourage everyone to use Slack to its full potential.
Yeah, great point. And I'm not sure if this is the one that you linked in, or if it was just the Waterbird Society, but I know the Waterbird Society also has its own kind of sub-channel within the American Ornithological Society that's like just for students to connect with each other and give each other advice.
Yeah, I need to find the updated link for that, but I think Juliet probably is the best person to message for the AOS specific channel, but it's also very cool. I don't have the ability to provide the link, but it's a very cool channel if you are interested.
Yeah, and maybe just message us on the Waterbird Society Slack for now, and we'll be able to get that to you at some point if you're interested in connecting that way. Anybody else have any comments or questions for now? Yes, Dave.
I just had a quick question.
Oops, I think you muted yourself.
Sorry about that.
There you go.
I just had a quick question, like a survey for all the presenters. Like, would you prefer, like for future meetings, would you prefer these lightning talks, or would you prefer to give a poster?
I think I prefer the lightning talks personally. I think it helps provide more presenting experience.
Yeah, I would agree with that.
Anyone else have an opinion or?
Yeah, I think the talks are good for giving presentation experience, but I do like the interaction of the poster a lot still.
Especially in person.
Yeah, definitely. This is great for Zoom, but if it goes back to being in person, I like the posters.
Yeah.
Cool, good points, thank you.
In the meantime, I also just want to say thank you so much for all of the presenters. I mean, for where you all are in your projects, they all sound really, really great. And it seems like you're on track, especially if there's no questions.
Thanks for moderating, Jenna.
Yeah, absolutely. Happy to. In the meantime, if nobody has any questions, I'll also plug the silent auction again. So all of the proceeds for the silent auction go for student travel and helping fund to get people to come to our next in-person conference. There's lots of great items and experiences like boat trips and paintings and really cool books in there. So definitely check that out. We have about 3 minutes until our next session. So maybe we'll just take a break and let everyone have a couple minutes to go get a glass of water or coffee or something. And we'll be back in 3 minutes and start at 4:20.
Thanks, Jenna and Riley.
All right, short break. We're back. And we're gonna get started. Is Henry here?
I'm here.
Excellent. You can go ahead and start presenting.
Just a moment. Hello, is anyone seeing? Is everybody seeing?
Yes, it looks great.
Okay, good afternoon. My name is Henrique Delfino, and I'm a researcher at Federal University of Rio Grande do Sul in Brazil. And I'm here to talk about our new project, Flamingos do Sul. So Flamingos do Sul, integrating ecology and ethnobiology in favor of the conservation of Chilean flamingos in southern Brazil. The flamingos are one of the most popular group of birds in the world, and in fact 3 species live in the southern part of South America. The Chilean flamingo, the Andean flamingo, the Puna flamingo usually breed in high Andean wetlands and move to lowlands during the non-breeding season, especially in the case of Chilean flamingo. During the non-breeding season, these birds often fly to the coast of Argentina, Uruguay, South Brazil, Chile, Peru. And in Brazil, in South Brazil, the most important area for these birds are the Lagoa do Peixe National Park. The Lagoa do Peixe National Park is an important conservation area for many migratory birds and for the flamingos. The park has a shallow saline lagoon, the Lagoa do Peixe Lagoon, and many different type of environments, from coastal ecosystems to natural grasslands. The park is especially important for flamingos because it's the only area in South Brazil where flamingos are seen all year round, especially Chilean flamingos. Despite this innegable importance, the Lagoa do Peixe National Park is a place of political and social tension right now, mainly due to the anti-conservation agenda of Bolsonaro administration, the president of Brazil, that puts the park under the risk of privatization and the flexibilization of conservation law in the area. But at local level, the community also makes a lot of pressure under the area with private interest to use the park as for tourists, for tourism, for fishing, for farming, and for livestock raising. Add to that the fact that the park is very poorly studied. Only few studies were made in the park in the last few years to support its importance and the importance of its conservation. Despite many birds that use the park, including the South American flamingos, are threatened and in populational decline. So it's in this context that the project, the Flamingos do Sul project, was created to unite the traditional scientific research with the community that lives nearby the park, the ethnobiology part of the project. So the goal of the project is not only to fulfill the ecological gaps, ecological information gaps that we have about flamingos in the area, but also use the full potential that this species can have to be an umbrella and flag species, not only for the conservation of Chilean flamingos, but also for the conservation of the park and for the other species that are associated with this environment. Among many activities in the project are the traditional ecological research and also the construction with the society of a management and conservation plan for the species in the area, the scientific communication, the science communication and environmental education to raise awareness about the park importance and the importance of the conservation of these animals, and the use of social media to raise awareness about the importance of Chilean flamingos and the conservation of the park. The project started in early 2021 virtually due to worldwide pandemic, but we plan that our field work will begin very soon in early 2022. Thank you for your attention. Make sure to contact us in case of any suggestions or any doubt. Sorry for my bad English, I'm still learning. Once again, thank you.
Thank you, Enrique. That was an excellent presentation. Looking forward to talking more about it later. Up next, we have Anna Zarn.
Yeah, that's me. I'll share my screen. Okay, hopefully everyone can see that. So I'll be talking about my PhD research on American oystercatchers in North Carolina today. So the region in the study is the lower estuary of the Cape Fear River within the coastal plains of North Carolina. This region contains a large majority of the state's wetlands and is also fed by two blackwater rivers characterized by low pH, high dissolved organic carbon, and low dissolved oxygen. It's also in the most industrialized watershed in North Carolina. And all of those characteristics together, so the presence of wetlands, low pH, low DO, industrialization, leads to an increase in methylation rates of mercury. And methylated mercury is actually the most toxic form, so that's why we're interested in that specifically. Mercury is naturally occurring heavy metal coming from things like weathering, wildfires, resuspension of sediments from storms, but it's also increased in the environment by things like cement production, waste management, and the burning of fossil fuels. So for the current study, it's part of a larger project quantifying mercury and other heavy metals throughout the food web of the lower Cape Fear River, since mercury does biomagnify as trophic level increases. But for today's talk, I'll be focusing on results from the American oystercatcher, which is one of our higher trophic level species, so it's probably going to have higher levels of mercury. And the main objective just overall is to quantify metal concentrations and get a baseline for the area, because it actually hasn't been studied very much, despite being kind of a biological hotspot for mercury methylation. So the American oystercatcher is a species of greatest conservation need, meaning that they are endangered or threatened, experiencing threats to life history needs. They're low in abundance and showing declines, all of which describe the American oystercatcher. So we monitored nesting pairs throughout the season, the summer of 2018 through 2020, and we actually continue that into 2021, but I haven't had time to analyze those samples yet. But we collected eggshells once they were hatched and breast feathers from chicks once they were big enough to be banded. And we also received feathers from birds in the Pamlico Sound to look at any spatial differences between the Cape Fear and another region in the state. So once we had those tissues, we analyzed eggshell membranes and chick feathers for mercury, and then carbon and nitrogen isotopes, which gave us more information on their trophic level and their feeding behaviors. And I can provide more information on the methodologies if you have any questions on that. So here are stable isotope biplots of our oystercatchers. The Cape Fear oystercatchers appear to feed at a higher trophic level because of their higher nitrogen signature. And that can definitely impact mercury bioaccumulation, since an increased trophic level generally leads to an increase in mercury as it does biomagnify. And here we see average mercury concentrations in our two tissues. The red bars are from the Cape Fear River, and the blue bars are from the Pamlico Sound, specifically Cape Lookout. And you can see we have a significant increase in the Cape Fear samples in both feathers and eggshells in 2019, but we don't see that same pattern from the feathers from Cape Lookout, telling us that there's something different between those populations in either their behavior or in the environment. And these levels may not be detrimental to the health of the populations as of right now, but any increased mercury concentration does have the potential to decrease successful reproduction, increase parasite loads, alter behaviors. So it's important to continue monitoring this over time. Some possible explanations for why we saw these differences include storm activity. Storms can resuspend sequestered mercury, and we did have a few strong storms leading into the nesting season of 2019. Immediately following Hurricane Florence in 2018, we saw elevated levels in shellfish, which remained elevated for quite a few months following, so that could have had an impact. An increase in dredging activity can also have the same effects as a strong storm by disturbing the sediments. Another factor could also be changes in the base of the food web. Since mercury biomagnifies with each trophic level, a small change at the bottom could result in significant differences in a predator. And based on some of our values in oysters in the Cape Fear River, there can be huge changes within a small time frame. I pulled out some values from October of each year, which is after the nesting season, but just to illustrate some of the differences we've seen. The average values are normally around 0.1 part per million, but as you can see, we saw very large fluctuations. Humans with 10 parts per million would be showing signs of mercury poisoning, so oysters with 20 parts per million is extremely high and very unexpected. So the extreme high values in 2020 were supported by other heavy metals that we were also interested in. And I'm really interested to see how this might affect values in American oystercatchers from this past season, which I have yet to analyze. But this definitely has the potential to reach potentially like the threshold for what American oystercatchers can handle in terms of heavy metals in their body burdens. So I'm definitely interested to see the results from that. I'll acknowledge everyone who's helped me with this project, and I look forward to any questions.
Thank you so much, Anna. And we're going to move right along to Dale.
Thanks, everybody. I'm going to rely on technology a little bit to kind of keep me hemmed into this wickedly tight format. And then I'm going to keep my chat open so I can answer questions real time if you post them in the chat. And then I'll leave a minute at the end for questions. One second.
Ecologists working in intertidal systems have long known that tidal dynamics control the temporal availability of resources for most animals in that habitat. The challenge has been in translating that temporal process into a variable that can be used in a spatial resource selection framework.
Although it's easy to see that tidal dynamics control resource availability, it doesn't mean that the pattern that results from that control is even across space. And you can see in this animation on the right, this map of islands in the Lower Keys, with the colors representing different amounts of time that habitat is available in a particular depth window. You can see that it varies across space, but also you see those differences in tidal fluctuations from day to day. This animation represents 1 month. 10 April to 10 May of 2017. So the pattern fluctuates in time, but it also fluctuates quite a bit across space. So we tested whether birds were selecting sites based on this time integrated resource availability. So these are sites that might look the same to you and I, that is, they have the same depth, the same vegetation types there, but they might differ quite a bit in how much time water is in a particular depth window at that site. And if so, then this would be a very underappreciated component of habitat quality.
We tested this notion in a habitat selection study of great white herons and little blue herons in the Florida Keys. The first paper was published by Kai et al. In 2018, and then Marisa Martinez picked up that work and expanded it up the Florida coast. In both cases, we found that the selection of sites by these species was strongly affected by this time integrated measure of habitat availability. In this case, we depict it in the figure with day averaged habitat availability for a site on the x-axis. And for example, if you can see that if you increase that habitat availability from 200 minutes to 400 minutes, the probability of selection of that site increases between 2 and 2.5 times. So both species are selecting sites that were shallow for long periods of time, relative to the average site in that area. Now, the attractive thing about thinking about time integrated spatial attributes is that because management is largely place-based, you can produce maps that managers can use. So in this case, we have these relative probability of use maps, and you can look at the one on the left, which is just an overall probability of use score from low to high. But you can also cleave off the low values and say, well, where are the places that are kind of our hotspots? And these would be like the core foraging areas where 70% of our observations are likely to occur. And you can get these sort of high priority maps. So when we looked across all of our habitat variables, we found that the time integrated resource availability was the strongest predictor of habitat selection for both our focal species. And the take-home point here is that it's really a small set of sites with the highest time integrated availability that are disproportionately important for foraging habitat for these two species. So when we couple these habitat models with, say, future models predicting sea level rise, it would allow us to prioritize particular areas for conservation based both on their predicted values now, but also under future scenarios.
Excellent, Dale. Thank you so much. I think that we're going to try to save all the questions for the end. So I think we might just try to move on to the next presentation, which is Helen.
Yeah, thank you. Okay. All right, so hello everyone. My name is Helen St. John. I'm a senior biology major at Grinnell College, which is in Iowa. But I actually am presenting today about this pilot study that I did over the summer in Rookery Bay National Estuarine Research Reserve, which is located near Naples, Florida, in Southwest Florida, where we were using game cameras to monitor disturbances of protected seabird colonies, and specifically black skimmers and least tern colonies. And before I dive into the project, I wanted to just start by thanking my mentors for this project, Colette Luzaw, Jeff Carter, and Nina Garfield, and then the NOAA Ernest F. Hollings Scholarship, which provided the funding for me to be able to complete the project. Okay, so there were 4 main questions that we wanted to answer over the summer. The first one was just if the Spartan GoCam game cameras can detect disturbances on Second Chance. And Second Chance is the sandbar island where these least tern and black skimmer colonies were nesting this summer. And the reason we're using these cameras specifically, which are the Spartan GoCam game cameras, is because they're wireless. And so they should allow us to monitor the island and the disturbances without us having to actually be on the island. And that sort of leads to the second question as to whether or not we can just in general use the cameras to monitor the colonies, because since we can't be out there 24/7, even if the cameras can't detect disturbances, they can at least sort of give us a better idea of what's going on while we're not monitoring the island in person. In terms of the disturbances, though, we do want to figure out sort of the type of disturbances that are most impacting these colonies and when they occur, because that will give us a better idea of how we can better manage the disturbances on Second Chance. So there were sort of 3 parts to the work that I did this summer. The field work, which was mostly just consisting of putting the cameras out there and then doing regular checkups on them for changing the batteries in the SD cards. And the cameras were put on our CWA signs, or critical wildlife area signs, which are located at these locations in the dots on the island, with red being where we put the cameras originally and the yellow where we moved some of them as the colonies moved throughout the summer. And then data collection was basically just me looking at all of the photos and videos that were taken from these cameras and recording this data about any disturbances that we saw. And so the disturbances were counted as disturbances if the birds were visibly flushing, or if there was something in the photo or the video that could potentially cause a flush, like a person or a predator. And then analysis was just some general data analysis in Excel and R for us to be able to answer the questions that we had about when and where and what type of disturbances were occurring. All right, so part one of the results, can they detect disturbances? We were happy to see that they could. Sometimes it was hard because, as you can see from the first photo, the least terns blend in really well with the clouds and the sand. But when you do look closely, you can see the least terns flushing, and probably because there's an osprey that's just landed near the colony. Another valuable thing was the cameras were able to record videos, so we could actually hear and sort of see the patterns of flushing when they occurred, even if sometimes we couldn't see what was causing the flush. And so this is a video of black skimmers. So that was fun to hear sometimes. In terms of just general monitoring with the cameras, we were pretty pleased with how the wireless component was performing, but sometimes it was a little bit clunky for us. The website, especially that comes with the cameras, only lets you look at 24 photos or videos at a time, and we had thousands of photos every day. So just using the website would not be sustainable. But the app that comes with the Spartan camera app was very helpful. And then in addition, we realized partway through that heat became a problem because it was corrupting the SD cards and some of the photos on the cameras. And so that just meant that we had to keep that in mind more in terms of how long we left each individual SD card out before putting a new one in. In terms of the types and when disturbances occur, when we look at how long each type of disturbance was lasting, which sort of gives us a better idea of how impactful they were on the birds, we see that unfortunately it seems like fishermen were impacting the black skimmers and the least terns the most out of the types of disturbances that we saw. And then in terms of the time and the days, we see disturbances mostly happening on the weekends, especially like Saturdays and Sundays, and then earlier in the mornings from 6 to 10 in the morning. And these two pieces of information are really valuable to us from a management perspective because they allow us to better target when our patrols go out to the island. Because currently we have a volunteer group called Team Motion that does patrols out to the island to look for disturbances and sort of prevent people like fishermen from getting on the island. But they don't really go out on the weekends, or at least they didn't this summer. And they also don't leave for the island until later in the day. And so they're missing these peak times and days of disturbances. And so with the information from these cameras, we're able to sort of direct their patrols better so that at the minimum, we're getting a better idea of what's going on in terms of disturbances on the island. And at most, they can help prevent some of these disturbances. And in addition, we were also just in general able to help law enforcement with some of the protection of this island because we were able to catch some trespassers on the island. Thank you.
Excellent project. Thank you so much, Helen. We're going to move right along to Jonah.
Okay, let me share my screen. Everyone see that?
Yes.
Okay. Thanks everyone for tuning in. I'm excited to talk to you about some of our ongoing research on saddlebill storks. So saddlebills are widespread in Eastern and Southern Africa, and they have a fragmented range in West Africa. And we really don't know a lot about them, even just basic things about life history, definitely nothing about population ecology, and nothing about movement. And these substantial knowledge gaps really prevent us from, one, understanding population dynamics in general, but also really knowing how the species is doing in order to assess status. And so aside, whoops, sorry, I just realized you guys said that. Aside from their striking beauty, saddlebills are really unique among storks and also I'm pretty sure among all birds, and that individuals have unique bill patterns that can be used to identify individuals. So they have this natural ID mark, which means we don't actually have to capture them. Sorry, we don't have to capture them to tag them for a population study. And so you can sort of think of this like using the spots of a leopard or a tiger to identify them by their coat pattern. And we've known this, or some people have known this about saddlebills since the early 1990s, but for some reason it's only been used twice over very short periods of time to estimate populations in Kruger National Park in South Africa. And so we initiated, as part of a larger study on saddlebills, we initiated a mercury site study using this method. And we started it in 2019. And our study area is in Western Zambia, specifically Lua Plain National Park, which you can see outlined in red there. Lua is in the upper Zambezi River Basin, and this area has highly seasonal rainfall with a wet season between November and April. And during this period, the grassland floods, and so everything you see here in this image would be underwater during the wet season. And then as the dry season comes on and progresses, water recedes and it remains in these ephemeral pools across the plains. And so we did this mercury site study the first 2 years in 2019 and 2021. And both years, they represented years of environmental extremes. So 2019 was the worst, just by coincidence, the worst drought in recorded history. And we only saw 15 individual storks that we identified, all of which were adults except 1, 2, or 3-year-old. And then 2021 was a year of above average rainfall where saddlebills were everywhere. In fact, aerial surveys found nearly 3 times as many this year than in 2019. And because of this abundance, it allowed us to develop the first aging guide for the species, which is going to be really useful moving forward because they don't reach adult plumage until their 6th year. And so now we're able to distinguish these cohorts. And you can see here, this is the age and sex structure of the 117 individual storks we identified in 2021, 73% of which were immatures, which immatures are in their 5th year and under. So that's interesting because this is all brand new information for the species. One of the most interesting things that we observed were congregations like this, where actually the largest group that I saw had 23 storks, which is one of the largest groups ever recorded in Africa. And what was interesting about this especially is that most of the individuals in these groups were immatures. 87% of the individuals that we identified in groups were immatures actually. And so because people don't really pay attention to the species, we don't know if this is uncommon, just because of the way that this species has been neglected and the way birds are reported in Africa. So kind of an interesting thing that we observed, and of course has created a lot more questions, but it kind of shows how useful this mercury site framework is going to be for long-term monitoring, because not only for like estimating population parameters, but studying these parts of their ecology that we know nothing about. And it also shows that this is a useful technique, not only just here in Lua, but it can be done anywhere where the species occurs and where people have cameras, really. So with that, I just wanted to thank the funders of this project, especially the Waterbird Society, and then African Parks for logistical help in the field.
Thank you so much, Jonah. And sorry, I realized I accidentally got the order of these 2 talks mixed up, but up next we have the talk on cat depredation by Helen.
Hi, everyone. Thanks, Jenna. I'm the Hawaii Conservation Coordinator for Pacific Birds, and so I'm just going to go through some of the work that we're just getting started in Hawaii related to cat depredation. Whoops. So just to set the context, Hawaii is one of the most isolated archipelagos in the world. There we are in the red rectangle, and as a consequence, we've got this really unique avifauna, and in fact, all of the birds that you see on this slide are endemic, but they're also threatened or endangered. So what's the problem with outdoor cats? They're obviously an introduced predator, and a good example of the issue is at the Hanalei National Wildlife Refuge, which is extremely well managed by the Fish and Wildlife staff there. But despite that, of 251 bird carcasses were identified as having been killed by cats. And that was over a 4-year period. 100 of those were the Ale Ula, or the Hawaiian common gallinule, and that has an estimated population of about 1,000 birds. And as you can see on the slide, the picture is similar for these very small populations of threatened and endangered birds. We're seeing high numbers of kills just at this single National Wildlife Refuge. And some of these were caught on camera too, as you can see. At the same time, we've got at least 20,000 feral cats outside on the landscape in Kauai. There's nowhere to take feral cats. The Humane Society has decided to become no-kill. People feed feral cat colonies outdoors, and people come from the mainland to do trap, neuter, release events. The conservation community is down with the trap and neuter part, but not so much with the release, especially because it has also happened near sensitive wildlife areas like Hanalei again at the top. So we wanted to try and make a change to how people viewed cats on the landscape. The current situation is that cats are everywhere. We've got wildlife dying. It's seen as normal to let your pet cat outdoors, and outdoor feeding is tolerated. We want to get to a situation where feral cats have been moved into closed sanctuaries. It's normal to keep your pet cat indoors and the wildlife is thriving. And we recognize that small businesses can help with that. They're mostly run by local people who care about the native wildlife and don't really want feral cat colonies nearby for health hazard reasons and because they recognize the damage that they cause. And part of trying to make this change is taking away some of the deterrents. So at the minute, people don't really know what to do to prevent feral cat colonies and feeding. It's difficult to do anything about it, and they're worried about confrontation and repercussion. So that was part of the target for our communications work. We also wanted to motivate people by making it easy to do the right thing, making them see that they belong to a much bigger group of people who believe the same thing, and giving them some tangible benefits. And we've been working with Brooke Tully to develop some of this work. So we developed, or rather Hub Osterlund of the Kauai Albatross Network developed the Wildlife Friendly Business Initiative to try and achieve these aims, and she brought all these partners in on that. It's very simple call to action. Basically, the businesses just need to have no residential feral cats on site, adhere to wildlife-friendly lighting guidelines, because we also have highly endangered seabirds on the island, which get grounded by lights and then eaten by cats, as well as run over and so on. And then for our native marine wildlife, not to release helium balloons. So it's a pretty simple ask. What's in it for them is they get a nice poster, they get a sticker, we will promote their business on social media, and we'll provide some of our volunteers to train their staff if that's helpful to them. And we're a very tourism orientated community, so this is a plus for local businesses. And we've just launched, so we're actively recruiting businesses. We don't have any results yet, but we're hoping this is one of the many steps that we have to take to try and move the dial towards thriving native wildlife here.
Thank you so much. Excellent presentation and really important work. We're going to move along next to Anna.
All right, hi everybody. I get situated here. Okay, so like Jenna said, my name is Anna Valerie. I work with Houston Audubon, and I'm going to be talking a little bit today about a project we started working on around September, so we're still deep in it, but in partnership with Audubon, Texas, and Rice University's Data to Knowledge Lab. And so the goal of this project is to develop machine learning to help better use unmanned aerial vehicle or drone footage for surveying waterbird nesting islands. So this group's pretty familiar with how colonial waterbird monitoring is typically conducted, especially at nesting sites. So most surveys are done with small aircraft, by boat-based surveys, or by walking through the colonies and attempt to get an accurate count. But each of these methods has its issues related to both disturbance and the amount of accuracy that you get. And so in the past 10 years or so, UAVs or drones have become a lot more prevalent in this effort, with a lot of success in using UAVs to survey single species colonies. More recently, cameras have gotten good enough and cheap enough to get species-specific identification from UAV surveys, but the digitization process of this footage can be really time-consuming and tedious. So development of automated bird detection algorithms is the next step. So this research was done as part of the Texas Colonial Waterbird Census, which is a decades-long effort to survey the entire Texas coast once a year, and a bunch of partners contribute data to this effort. And I'm specifically talking about data that was collected at 2 colonies, Chester Island, which is in Matagorda Bay, and North Deer Island, which is in Galveston Bay. And so this is kind of building on some work that I've been doing with Houston Audubon over the last couple of years, surveying one of Galveston Bay's colonial nesting islands. And we've had a lot of success. So UAV surveys allowed surveyors to see almost 10,000 laughing gulls in 2019 and 5,500 in 2020 that were missed during the boat-based surveys. And the same, we had similar results for both the mixed tern species and also our brown pelicans. But it takes nearly 40 hours of labor to digitize birds, all the birds on a 6-acre nesting island. And the 2 islands I just showed you are over 100 acres each. So these time and resources are what most conservation organizations don't have time to dedicate to for the sake of more precise and accurate counts. And so to try and address this issue, we partnered with the Rice Data to Knowledge Lab to develop machine learning and computer vision algorithms for the detection of birds using images that we took with our drones. Our 2 main objectives are to not only count and survey the number of birds, but also to train the computer to identify the birds to species in these surveys. And so we had 15 main species identified across the footage so far. We're really going to focus on brown pelicans, laughing gulls, and this mixed tern group, which eventually our goal is to separate out. It's mostly royal and forester's terns, to train the algorithms to separate these species out. But for now, we're grouping them together and focusing on identifying all the other species. Also point out, we are training it to identify trash and debris on the islands for hopeful removal later. And so the first step is to develop a simple object detection algorithm for the water birds seen on the 2 islands. And object detection is really a combination of the localization along with an automated image classification. And so this is super complicated, but if anybody has questions about this, I can put you in touch with the Rice lab. But so the results of our first model were really great. So the first model was a bird-only detector. And this is so we could learn how well a convolutional neural network-based object detector could detect just birds in general. And overall, again, it does a really excellent job with an average precision of 93.7%. Which is kind of to be expected because we've seen a lot of successful other papers using this on like penguin colonies and other single species colonies. The second step was to try and train a model to identify bird species, so create a bird species detector. Specifically for the 3 most annotated classes, so that brown pelican, mixed tern, and laughing gull class. All the other species for this initial goal were lumped into other. Eventually, we will break them further out. And so at the bottom, you can see those average precision metrics, and they're not as good as the bird-only detector. In particular, the other class, which we are working on, was obviously not very good. But even 80% for laughing gulls is a little unfortunate for what we would like to see. And so even though this model was initialized using the original bird detection model, it's unlearned how to correctly localize the birds once we added in the actual species ID aspect. So our team's already working on addressing these challenges by exploring a few different options for improving the algorithms, but it is challenging once you throw in multiple different species. So successful code and training algorithms really could help identify thousands of images in a relatively short amount of time, much shorter than the 40 hours it took to identify images from 6 acres. It could also increase accessibility of this technology for use by partners across the state or elsewhere. And so our work is ongoing. Again, we just started in September with the goal of creating accurate and useful algorithms for applications across the Gulf of Mexico and into other ecosystems. And our next steps include continuing to improve that species ID algorithm and eventually providing, hopefully, open source codes to everyone. So for whatever reason, this is like zoomed in weird, but special thanks to all of the collaborators at Houston Audubon, Texas, and with the Rice Data to Knowledge class. I'm not an expert in developing the code, but if you have questions about how we're doing this, I'm happy to have you email me and I'll connect you with the correct collaborator.
Thank you so much, Anna. And we'll move on to our final speaker, Graham.
All right, hello, I'm Graham Good, and I'm going to be talking to you guys about the response of the relative abundance of fire ants to coastal island restoration and vegetation. But I will tie it back to birds. Hello? Okay, so the Gulf Coast barrier islands of Louisiana provide crucial nesting habitat for many wading bird species, shorebirds like Wilson's plovers and black oystercatchers, in addition to seabirds such as brown pelicans, black skimmers, and several tern species. And these islands also provide excellent habitat or excellent opportunities for foraging, as all the birds can rest on these islands while they forage in the larger area. So because of the importance of these islands for avian species, there have been many restoration projects on numerous islands with the goal of maintaining or creating habitat for them. And so these islands, the two islands pictured here, are Queen Bass Island on the top and Raccoon Island on the bottom. And these are both off the coast of Louisiana, and we focused on these two islands in our study. And these images show the dramatic changes that have occurred as a result of the restoration projects. So in the top photos, you can see on Queen Bass Island in the most recent restoration project, the use of imported dredge material used to fill in that large open area in the 2018 pre-photo, and then it's filled in the post photo. And this is typical of Louisiana coastal restoration because it increases both island size and area. And so one animal that may be taking advantage of this restoration is the red imported fire ant. And so either through natural dispersion or potentially as a consequence of this restoration activity, the red imported fire ant has colonized several of these coastal islands. And predation of fire ants on nesting seabirds have been studied on barrier islands off of Houston, Texas. But besides that, most of the evidence is observational, and it's been mostly on brown pelican chicks and eggs. But this has sparked interest into the dynamics of these fire ant populations and how they impact nesting seabirds. So our objectives were to collect new relative abundance data on Queen Bass and Raccoon Island, and then compare this to the 2018 and 2019 counts, and then to also explore the possible impacts that island restoration, sampling date, and habitat have on fire ant relative abundance. So we wanted to see if our independent variables were good predictors of higher fire ant counts or lower fire ant counts. So to acquire the fire ants, we created transects that were 50 square meters in area, and they consisted of 3 rows of 9 tubes seen in the bottom right image. We baited those tubes with Vienna sausages, and we spaced them evenly 5 meters apart, and they were left for 1 hour before collection. And then we preserved the ants in ethanol and counted them. So we found that habitat and thus vegetation had strong predictability for fire ant density. This is looking at the top left figure, and with marsh habitats yielding higher counts than dune habitats. But despite this relationship between habitat type and fire ant counts, it gets complicated by restoration status of that habitat. And this is the top right figure. So you can see that Queen Bass has higher counts in the unrestored sites, while Raccoon Island had higher counts in the restored sites. And so this is important to note that while there are similar trends in some variables like the habitat counts, it gets complicated because of the interactions between these variables. And in this case, that's the habitat type and the restoration status of that habitat. And then we also found that sampling date had some predictability for greater ant counts. And this suggests that factors, including storm surges or local events, can alter fire ant activity or population size on a shorter time scale. And then with respect to the historic versus current counts, Raccoon Island had higher counts in 2021, while we found higher counts in the 2018 2019 data on queen bass. So there's a lot of inconclusive results. So with all that being said, there's definitely a need for more research. Although these ants have been observed predating on seabird chicks, we don't know the extent to which they impact these seabird communities. But we do need more data before we can draw larger conclusions on their role in these island ecosystems. Important to note is the uniqueness of these islands. Their locations and restoration strategies result in differences that are important to consider when predicting these fire ant abundances and how they affect the associated avian communities. And then just to acknowledge the NSF, the University of Louisiana at Lafayette, and my home institution, the Evergreen State College. Thanks.
Thank you so much, Graham. All right, now we have some time for questions for our speakers. For any of the speakers in this session, I'm going to go back and read out some of the questions that were in the chat as they were asked. This first one, I think, is for Anna Zarn. The differences over time are interesting, especially the peak in 2020. Any anecdotal thoughts about impacts to American oystercatcher hatching success and or chick survival?
Right, good question. So anecdotally, we didn't see a difference in hatching success or chick survival between the years, even though we did have a higher concentration within 2019. We did collect some eggs that were unsuccessful in hatching to test those for concentrations as well. So it'd be interesting to go back and look if those were significantly higher in mercury. Maybe that led to why they weren't successful. I think all of the values of eggshells we had were around 0.1 part per million. And I think the threshold is anywhere between 0.5 to 0.8, depending on the species. So I think we're pretty safe to say that they're under the lethal limit as of right now. But it would be interesting to go back and see if those were significant or not. So.
Yeah, great. And then another question for you, does mercury sequester in dietary calcium sources for eggshell formation? And if yes, could the type of tissue used, eggs versus feathers, affect inferences regarding mercury uptake?
Right, so birds use two main pathways to get rid of mercury. If they're female, they'll shunt it into growing eggshells and growing feathers. So those are the main two ways to get rid of it, while they're to get rid of their internal organs. But they do generally correlate to one another. So the concentrations within the adult will be similar to the concentrations within like an eggshell or a feather as they're grown. I think the thought to include both are kind of as a complementary source. So we'll see a similar pattern in the eggshells to the values in the chick feathers, since they're grown at a very similar time. So the concentrations are normally complementary. I have to look at maybe timing differences. Maybe it does tell us a little bit different, but in general, it's more of a complementary stat. So.
Good, thank you. Our next question is for Helen St. John, I believe. Was there any indication that potential predators perched on your camera posts?
No, we had, so the CWA posts had like the big spikes on the top, so we didn't have any predators landing on it, but it was possible that they could be like behind the posts and then we wouldn't see them on the cameras. Yeah, and I think there's a lot of questions directed at my project, so I can just,
yeah,
I can read them if you want. Okay, so the next question was from the other Helen. Did cameras have issues with rain or humidity? In terms of like the being able to take photos, we didn't really have issues with that. Sometimes the clarity of the photos were a bit of an issue because there was a lot of rainy days over the summer, and sometimes there were raindrops on the lens, but generally that didn't prevent us from being able to like determine like flushing or disturbances. And then on the app, we were able to look at all of the cameras, all of the photos and videos at one time. And we could download them from the app as well. Okay, Jeff Costa, the batteries lasted about a week, but it depended on the settings we set for the camera. So how often we set them to take photos, or whether they were like motion sensing or not. This summer was sort of used a lot to like play around with those settings to see what would work best in terms of being able to detect what we thought was going on out there, while not like requiring us to change the batteries every day. And so we got it to be about a week before we had to change the batteries. And then the data was transferred wirelessly with the, like, I think it was the AT&T phone like network, like we used, that's what the cameras are like connected to. And so I guess in terms of being able to look at the photos on like an app or something, we had to be able to connect to those wireless networks. Okay, 2 from Alex Smith. The island that we looked at, Second Chance, is pretty small. So we were able to see, with the way the cameras were set up and the way the signs were spaced out, we were able to get a pretty good view of the whole horseshoe shape of the island. But there weren't colonies everywhere on the island, so we didn't look everywhere on the island because we wanted to focus specifically on the colonies. And it would, yeah, if the islands were larger, we would need either more cameras, or it would just be harder to collect data with the cameras. I think this is the last question for me. Looking at, we were only, so the question is, were you just looking at frequency and disturbance and duration of the disturbance, or did we collect data on the distances at which the birds responded to disturbance sources? So because this study was sort of just trying to see if the cameras were even like a good resource for Rookery Bay to use to monitor disturbances, we were really only focusing on whether or not the disturbances occurred, and if we could tell like how long they lasted and what was causing it. So we weren't able to get to anything in terms of like the distance between the disturbance source and like when the birds decided to flush. Yeah, I think that's everything.
Great, thank you so much for going through all of those. Up next, I see a question. There were some that were for Dale that were answered directly in the chat, and same thing for Jonah. Thank you for answering those. This next one is for Helen Rain. Have any native mammal species or other been seen predating on the cats?
Yeah, we don't have any native mammals apart from the Hawaiian monk seal and the Hawaiian hoary bat. So that's one of the problems. There's nothing to predate them, and we have a fantastic mild climate all year round. So there's really nothing that takes the cats out in the kind of numbers that you would need to have any kind of control method, and just huge resistance to any kind of control from a vocal minority.
Yeah. Another question for you. Has anyone looked at a feral cat birth control protocol? Alex Smith said that he had seen some deer birth control protocol in place on some islands in North Carolina, and was wondering about using it there.
Yeah, that's an interesting idea. I asked my colleagues about this, and apparently they have discussed it, but the challenge was that if it was an oral bait, they had to keep consuming it for it to be effective. But Alex, if you know of other ways of administering it that might work for cats, we'd definitely be open to thinking about that.
One more question for you that I can see here. On Long Island in New York, we have a big problem with feral cats and birds. Do you think that there's a way to adopt the wildlife friendly business initiative to a person-to-person neighborhood scale?
Yeah, I think that definitely is. The wildlife friendly business is kind of one tool in the arsenal. But before we started that, we were working on a kind of cat safe, wildlife safe Kauai, with the idea of, again, asking people to keep their cats indoors because that's safest for the cats and also safest for people. And I mentioned Brooke Tully before. She runs a kind of communication course that helps you design the behavioral journey you want to take your audience on. And we're going through that process with her now. So I'll have a look who asked that question, and I can share some of the outputs from that, because I think it is really transferable to other places.
I think it was Ariel Santos.
Cool, yeah.
Yeah, thank you. And I think that the next couple questions are for Anna Valerie. Anna, Alex Smith asked, have people seen much direct mortality from birds being disturbed by a drone and then colliding with the drone during disturbance response? And at what height do you usually fly the drones for your surveys?
Two-parter, cool. Yeah, there is not a ton of evidence, I should say, for water birds, there isn't a lot of evidence of birds getting spooked and then hitting the drone. If anything, the challenge is birds getting spooked and then their nests being vulnerable for predators, including the laughing gulls, who are just waiting there to snatch up other chicks, especially the tern chicks and on a few colony skimmers. But part of my master's work was to try and develop like what height was ideal for flying at. And so we take a really safe 50 meters, which has, and I can show footage if anybody wants to see it. I'm happy to share some footage, but at 50 meters, the birds don't even like orient them. Like they don't even look at the drone. And the sound of the drone is something we consider too. The decibels that the drone, the smaller drone, it's a DJI Phantom. For this work, we were using a bigger drone, but it's the same thing. You can't hear the drone over the sound of the colony. So it actually is, the birds do not notice, or they show no indication of noticing. So we don't have any issue with that. And so yeah, 50 meters was with our DJI Zoom. And then, but 50 meters is also what we use for a slightly larger drone, but it was a larger, quieter drone. And things are just getting better and better as far as technology goes. So I think the drones are going to continue to get quieter and more stealthy as we can continue on.
Yeah, thank you. Another question for you. How do you verify accuracy with dark plumaged water birds that nest below the canopy?
Yeah, so this isn't the islands we're looking at are primarily vegetationless. Most, I guess, Chester Island, as opposed to North Deer. North Deer has a little bit of vegetation in them, or trees. I should say they have trees in the middle. Chester has more. The drone can, and the footage, the AI can pick up the dark plumaged birds on the top of the trees, which is where the majority of them are kind of, especially these water birds. The great blue herons are the ones that I'm thinking of, nest kind of towards the top. If there are birds nesting like deep in the trees, we aren't capturing those with the UAV. Clay Green, I know, had a student doing work kind of on observations of this that I heard maybe a year or two ago. So he might be a good person to reach out to about that.
Yeah, great. And another question, Dave asked, have you tried to estimate nests or breeding pairs? And do you think it's possible via machine learning, or would you have to do that manually?
Yeah, no, that's a great question, and something we've talked a lot about, both as the Texas Colonial Waterbird, just from doing our boat-based or ground-based surveys, and with the drone. So we make the assumption, I think, that an adult on a nest is one nest. And so if there are two adults on a nest, it may double count. And when we ran the AI, and this is kind of the problem for, it's not just the AI problem, it's also human error of if you're able to like determine what is a nest, especially when there's thousands and thousands that you're trying to count. But the AI, interestingly enough, would identify like 3 pelican chicks. We had an example of 3 pelican chicks, and it identified it as one nest. So I think we can set it we basically train it how we want it to ID things. And so we're currently training it to identify adults. And we are going to set boundaries, because for all these islands, we pretty much assume anything within the breeding area is and it's an adult is likely on a nest. And then all the loafing adults kind of hang out towards the edges of these islands. And so we are going to exclude them after the fact.
Yeah, great. Really, really interesting. Jonah was just asking if you're familiar with the automation research at the University of Florida on wading bird colonies.
I saw that question. I'm not super familiar with their work. We did reach out to Duke University and a university in Australia to check in with them about what code they were using for doing similar AI developments, but I'm not sure. My colleague Hank Arnold with Audubon, Texas is actually the one who's super knowledgeable of all the projects going on. So I'll have to ask him if he's familiar with UF.
Yeah, and you should reach out to them. It's Morgan Ernst's lab, and I've worked for them, and they've been doing it for a couple of years. Same stuff with different species.
Yeah, that's it is interesting how the different habitats and species can really make things. This one, our goal is really, the main goal is to create open source algorithms for the Texas colonial waterbird participant, so all the organizations doing those surveys, with the hopes that eventually you could potentially beef it up to other ecosystems and species. But yeah, I'll definitely reach out to her. Thank you.
And I think one last question for you. Do you think the future of drones would be able to differentiate between juveniles of different species, or even differentiate between distinct juvenile plumages with the same species?
Yeah, I do. I think it's less so the AI, it's more so the camera, how good your footage is. And so we're very lucky. Hank Arnold with Audubon, Texas had a stellar camera on this, really on a larger drone. And so we were able to get massive photos. And so the goal is to eventually, like I said, train to identify between those two tern species, which we almost can't do from the boat. But I think that once we can program like the varying plumages, it should be able to pick that out, no problem. It just depends on how high quality images you're able to get at that 50-meter safe flying height.
That's amazing. It's so exciting.
Yeah.
Yeah, thank you so much, Anna. I think I have a couple questions for Graham now. First, do you notice any differences in depredation by the ants on chicks that are precocial versus semi-precocial versus altricial?
No, because they don't really know much about the ants on these islands. So that's why we were kind of trying to get the relative abundance data to hopefully do stuff like that later, I think is the idea. But I have no good answer for that. Sorry.
Yeah, thank you. And another one, are there any control methods being trialed for the ants?
Not that I know of on the islands. Because again, yeah, people aren't really focused on them yet. There's been a couple studies, like I said, in Houston, Texas, but yeah, again, no, they're mostly focused on the birds out there.
Okay, great. And I think that those are all of the questions that I saw in the chat. Does anybody have any lingering questions for any of our speakers, both from the first or second session? You can go ahead and just unmute yourself, give it a moment. If not, I just want to say thanks again so much for all of our speakers. These are great presentations. I know it's so hard to fit all that information in 5 minutes, but very well done. And thank you again to my technical assistant, Riley, for helping with putting this together. There is a virtual field trip happening right now, I believe. So if you have nothing going on, definitely hop on over to that. I think we're going to rice paddies in Japan today. And again, check out the silent auction, bid on some things. Thanks again.